Hello from New York to everyone watching from around the world. Thanks to Peter for reaching out to me to give this talk. Thanks to the organizers for having me. And most of all, thanks for giving me a chance to talk about Mycangia. I did my PhD work on ambrosia fungi, their evolution, diversity, and relations to Mycangia. And I came away from that thinking that Mycangia were extremely interesting, but unfortunately underappreciated organs. So this talk is a brief summary of an upcoming chapter that I've written with Tom Harrington and Peter Biederman on Mycangia, what we know about them, what we don't know about them, and why they're so critical to the evolution and maintenance of ambrosia symbioses. Now, ambrosia beetles aren't the only animals to have developed agriculture. They share this distinction with fungus-forming termites, fungus-forming ants, and the single species of fungus-forming primate, Homo sapiens. But what distinguishes them from these other farmers is that they need to generally move to new trees every generation and start entirely new farms, and that they rely on Mycangia. You see, ambrosia beetles farm in the xylem of trees, a traditionally nutrient-poor substrate, but there they farm ambrosia fungi. These are specially co-adapted cultivars that have become so good at being food that their spores can no longer disperse independently. They entirely rely on ambrosia beetle dispersal to move to new substrates. Ambrosia beetles fly to new trees, typically every generation, and cultivate the same ambrosia cultivar that they were raised on, despite the wide variety of other wood commensal fungi and other microbes, both in their tunnels, on the surface of wood, and elsewhere. How do they do this? The answer is pockets. Just like the pockets of a jeans or the pockets of a backpack, pockets come in a variety of sizes in ambrosia beetles, and the size and type of pocket has a large effect on which fungi the beetle can take along with it. But what do these pockets really look like in ambrosia beetles? If we take this beetle apart, for example, this is Xylosandrus crassiusculus, we'll find a conspicuous pouch or pocket-like organ just in the pronotum. And if we were to place this organ on a microscope slide, stain it, and squish it under a cover slip, we would notice it was filled with a pure culture of fungal propogules. This is the ambrosia fungus that the beetle is going to farm when it flies to and establishes a new gallery. Let's back up a bit and talk about how Mycangia were discovered. The word ambrosia comes from the Austrian monk Joseph Schmidberger, who noticed in 1836 beetles feeding upon a white, salty, he apparently tasted it, substance that he figured was derived from the tree sap. It was Hardig who later realized this was a fungus, but assumed it was spontaneously generated inside the beetle's galleries. Henry Guernsey Hubbard realized that the beetles actually introduced the fungus purposefully. And for decades after that, the assumption was this was done either superficially with fungus spores on the surface of the beetle or through the gut with fungus propagules that were eaten and could survive passage through the beetle until it arrived in its new gallery. But it was Helene Franke-Grossman who had the intuition to realize that it was quite unlikely that ambrosia fungi could travel only on the surfaces of these beetles and yet maintain the extensive associations that ambrosia beetles rely on. Drawing upon her own experience in wood-associated insects, especially sericid wasps, inspired by Paul Buchner's discovery of microbial carrying organs in many insects, and noticing Heydorn's observation that overwintering Tripodendron beetles had completely empty guts yet still managed to transmit and inoculate ambrosia fungi, Helene Franke-Grossman hypothesized that there were special organs inside the beetles' bodies that might be for the purpose of transporting fungal spores. And she sought out to dissect and discover them in many beetles, and she was correct, discovering many such organs that she proved were full of active living fungal spores. She would go on to discover multiple such organs, not only in her 1956 discovery, but in the next decade plus, leading up to her 1967 foundational review of the symbioses. But she never really had names for them at first. She originally called them cuticular glands or folliculose glands, But in 1963, the mycologist Lech Batra introduced the term Mycangia to refer to the growing number of these structures. Franke Grossman would later suggest and prefer the term Mycangia, which is probably more grammatically correct, but it never really overtook Mycangia in common use, especially in the English-speaking world. For an interesting discussion on exactly this topic, check out this paper released by Vega and Biederman this year, which discusses the two terms and their merits. I should mention that for the purposes of this talk, I'm only talking about glandular mycangia, even though the term has been applied broadly in the past to include structures such as hairs or small pits that spores might become temporarily stuck to or lodged in. But all glandular mycangia share a few key features. First, they're typically invaginations of the cuticle that form vessel-like cavities with small openings. This is why ambrosia fungi are considered ectosymbionts, despite the fact that the organs appear to be inside the beetles' bodies. Glandular mycangia are all associated with gland cells, either lining the mycangium itself or placed nearby, that secrete into the mycangium lumen, and they contain fungal propagules that enter and exit through that same opening. In the time since Franke Grossman, a wide variety of mycangia have been discovered across ambrosia beetles. I don't have time to discuss each one separately, but I do want to impress the fact that these mycangia are all extremely different, not only in where they are on the beetles' bodies, but their size and their shape. Each of them separately evolved for the purpose of carrying and maintaining ambrosia fungus propagules. We've come a long way in how mycangia are visualized to show us what these organs look like, from the hand-drawn illustrations of the early workers to paraffin-embedded cross-sections, as shown here in G, to microcomputed tomography, or micro-CT, which can give even clearer cross-sections as in E, and can even produce intricate 3D models of the size, position, and shape of mycangia. Mycangia can also be dissected and imaged using microscopes, and combined with the 3D structures produced by micro-CT, this allows us to get a true picture of what mycangia really look like that can't be fully communicated using cross-sections. Mycangial propagules come in one of two flavors, either yeast-like, which bud and divide like true yeasts, or thallic arthric cells that grow a bit like hyphae that form septa and then break apart and disarticulate into individual cells. But in either case, these are dimorphic forms of filamentous fungi. When ambrosia fungus propagules enter the mycangium, they switch to this mycangial form, and when they exit and inoculate beetles' galleries, they switch to a filamentous form, growing and filling the gallery walls and eventually sporulating, producing the spores that the beetles will feed on. The conspicuous gland cells associated with mycangia have been noted since the earliest observations of mycangia, and these gland cells have been described in detail. Here's an example of a gland cell associated with a single small pronodal pit mycangium of a platypodine. The gland cell is connected to the pit with a small channel through which its secretions pass. Here's an example of a single gland cell embedded in the wall of a larger mycangium, here in the bark beetle genus Androctonus, but very similar in form to the gland cells embedded in the mycangium walls of many of the larger ambrosia beetle mycangia. What's not known is exactly what constitutes the secretions produced by these gland cells that's secreted into the mycangium lumen. What is known is that those secretions support the selective growth of specific co-adapted ambrosia fungi. This fact is well supported in the literature, though not necessarily in one place. Here's a few important stops along that path. In 1969, Schneider and Rudinsky reported seasonal changes in gland cells of the mycangia of Nathotrichus. The gland cells were small and deactivated while the beetles overwintered, but when exiting hibernation, the gland cells activated, became larger, and began to secrete into the mycangium lumen once the beetles started to take fungal propagules into their mycangia. Schneider followed up on this in 1975 by making observations in multiple different ambrosia beetles and coming up with a generality that gland cells didn't activate until ambrosia fungus propagules had entered the mycangium, at which point the gland cells activated and began to secrete. In turn, the ambrosia fungus propagules switched to mycangial forms and began to propagate. More recently, in the excellent 2018 paper of Skelton et al., mycangia-mediated selection was demonstrated experimentally and showed that types of mycangia can have not only broad specificity to genera of ambrosia fungi, but can have even more specific specificity to different species or strains of ambrosia fungi within those genera. The results of these studies and the rest of the literature reveals a Mycangium cycle that is essential to the ambrosia symbiosis. When ambrosia beetles emerge from pupa as young adults, their Mycangia are empty. But as you can see in the gallery shot above, ambrosia beetles live in very close proximity to the thick ambrosia growing along their gallery walls. Through a variety of passive methods, such as simply scraping ambrosia fungus propagules as they pass, and active methods, such as doing special behaviors, dances, jigs, or even averting their Mycangia and pulling them back in, ambrosia beetles non-selectively take ambrosia fungus propagules, as well as whatever other debris and fungi are in the region, into their Mycangia. Once these propagules enter the Mycangia, along with other propagules indicated here in red as opposed to the ambrosia propagules in blue, the gland cells respond by activating, growing larger, and secreting secretions here in yellow into the Mycangium lumen. In response to these secretions, the ambrosia fungus propagules begin to switch to a mycangial form and propagate. Eventually, they fill the entire Mycangium lumen with a pure or nearly pure mass of fungal propagules, pushing out any non-adapted spores and debris into the external environment. Beetles then, at this point, fly and leave the gallery to establish new galleries elsewhere, during which the Mycangium overflows with fungus propagules. As the beetles spore, these overflowing fungal propagules are inoculated onto the gallery walls, germinate, and begin to grow in a filamentous form along the wood surface. Eventually, the inoculum within the Mycangium deteriorates as the gland cells deactivate and stop secreting, and the ambrosia fungus has already established on the wall. Eventually, it will sporulate with conidiaphores producing spores that are both nutritious and ready to enter Mycangia for the next stage. The next generation of beetles, when they emerge from their pupa with empty Mycangia, will start this entire process over again. And we can appreciate this as a Mycangium cycle where the fungus consistently cycles from growing filamentous on the gallery walls and sporulating to growing as a yeast-like or phthalic arthric state in the Mycangium lumen. This process repeats itself over and over again and is how ambrosia beetles are able to bring pure or nearly pure inoculum into their new galleries. I say nearly pure because it's clear that there are some fungi that have become adapted to persisting in Mycangia and being transmitted along with ambrosia fungi whose role isn't yet known completely. These include yeasts as well as filamentous fungi. But what is clear is that ambrosia fungi both dominate the growth along the gallery walls and the thick ambrosia beetles feed on, as well as the Mycangium propagules that typically fill Mycangia during flight and gallery initiation. In the ambrosia beetle phylogeny shown here from Holzer and Selinsky's 2017 review, note that every blue clade is an ambrosia beetle clade. Ambrosia beetles have separately evolved multiple times from bark beetle ancestors, meaning that they separately took dives from the nutrient-rich phloem, feeding on both phloem tissue and fungi, down into the nutrient-poor xylem below, relying on fungus farming to extract the nutrients they need. Mycangia appear to be prerequisites for this shift because every clade of ambrosia beetle seems to have originated with the development of at least one type of Mycangium. It appears that an organ that allows for the selective maintenance of specific fungal strains, rather than relying on superficially attached fungi, is required to start true ambrosia symbioses. And here I should mention that some bark beetles have Mycangia. Some of them are rather simple and not that selective, but some of them, such as the Mycangia of the bark beetle Dendroctonus frontalis, are just as large, developed, and selective as the largest of ambrosia beetle Mycangia. But these beetles still live in the phloem, and although they rely on fungi as a large or majority of their diets, they do not farm fungi in the xylem like ambrosia beetles do. Perhaps this is an example of an ancestral state of a beetle that has adapted everything it needs to dive into the xylem and farm fungi, but hasn't done it yet. Once a beetle lineage has developed Mycangia, probably initially to carry only facultative fungal symbionts, that Mycangium may facilitate the true domestication of those cultivars into domesticated crops. All crop domestication requires that selfish evolution be reduced or eliminated in the crop, lest it develop traits more conducive to its own survival and dispersal than being a good crop for the farmer. And all farming animals take steps to limit genetic diversity in their crops, usually in long-lived forms. These processes are well documented in fungus-forming termites and fungus-forming ants, but not yet described in ambrosia beetles. The genetic bottleneck inherent in the Mycangium cycle is most likely critical to this process in ambrosia beetles, because only a few propagules enter the Mycangium and then fill the Mycangium due to asexual budding or propagating of propagules, and are then the nearly genetically uniform inoculum used to start new galleries in the next generation. Taken over a long series of time, this leads to frequent genetic bottlenecks, which might be conducive for the true domestication of ambrosia cultivar. This is further backed up by the fact that the origin of Mycangia appear to predate or closely match the origin of their associated ambrosia fungi, suggesting that functional Mycangia need to stabilize before they can domesticate lineages of ambrosia fungi. This appears to have led to patterns of association between the different lineages of ambrosia beetles and their different Mycangia and different types of ambrosia fungi. A pattern first recognized by H.G. Hubbard more than 100 years ago, who noticed that more closely related beetles formed more similar-looking fungi. But it was Richard Roper who in 2011 reported that these associations might be more closely linked to Mycangium type, and more specifically, Mycangium size and shape. At the time, there were two genera of ambrosia fungi, and Roper realized that Raffaellia appeared to be associated with smaller and less complex Mycangia, whereas Ambrosiella was associated with larger and more complex Mycangia. This insight became the inspiration for my thesis and is a topic that's quite close to my heart. Raffaellia is indeed associated with many different types of Mycangia and many completely unrelated ambrosia beetles, and different Raffaellia species can co-inhabit Mycangia at the same time. On the flip side, Ambrosiella and the four related genera in the Stratocystidaceae that are all ambrosia fungi are each associated with a different type of Mycangium and a different lineage of ambrosia beetle, and tend to have strictly pure inocula of propagules inside of those Mycangia, never being observed to share Mycangia with another Stratocystidaceae ambrosia fungus. In the time since, a number of new ambrosia fungus genera have been discovered, all with different patterns of association with ambrosia beetles and their Mycangia, and I'm excited to see what new ambrosia fungi appear in the future. But what's clear is that Mycangia define ambrosia fungus cultivar. So we've decided that Mycangia define which ambrosia fungus cultivar that an ambrosia beetle will farm. This is important because the many unrelated ambrosia fungi convergently arrived at being good crops for ambrosia beetles, and likely have completely different ecologies with consequences for the ambrosia beetles and the symbiosis as a whole. But ambrosia beetles aren't all the same either. Fungus farming evolved more than 10 separate times from bark beetles, potentially from different ecological backgrounds and potentially with different ecological consequences or strategies for farming. Therefore, each beetle Mycangium fungus trio should be considered a unique symbiosis, and conclusions made on only one trio should only be carefully applied to others, since their ecologies and biologies might differ substantially. So really we should say that Mycangia define ambrosia symbioses. In conclusion, Mycangia are diverse, active, and interesting organs. The Mycangium cycle is critical to ambrosia symbioses. Mycangia drive co-adaptation and domestication of ambrosia fungi. Every beetle Mycangium trio is unique, and that's important to keep in mind. And Mycangia are excellent model systems for symbiosis, because there's still so much we don't know about them. For example, there's still whole lineages of ambrosia beetles for which their Mycangia have not been discovered yet. We still don't know what it is that gland cells secrete, and we don't know the molecular mechanisms of Mycangium-mediated selection. For example, do the Mycangium glands produce some nutrients that only ambrosia fungi are tuned to, or do they somehow change the microenvironment of the Mycangium so that it's more conducive to ambrosia fungi? We don't know what triggers gland cells to activate and deactivate, and we don't know what triggers ambrosia fungi to switch from filamentous to mycangial states and back. This is where I'd like to plug the Bark Beetle Microbiome Research Coordination Group. This is a worldwide group of bark and ambrosia beetle symbiosis researchers who are working on tackling these and other questions related to the symbiosis. The meetings of this group, which happen multiple times a year, are open to the public via Zoom, and I encourage anyone who's interested in bark or ambrosia beetle symbiosis to participate in these meetings and to come and join us. It's an open and friendly group, and I'm really excited to see what gets discovered in the next few years related to these and other questions. So, with that, I would like to say thanks so much for your attention. Thanks for having me, I'm happy to take any questions either now or later through email about Mycangia, and stay safe, everybody. Thank you very much.